Hi, my name is Amir Manzour and I'll be explaining the following analysis. As we know, RNA molecules perform various functions based on their conformational specifics, and these molecules can have evolved into structures that can stabilize into complexes by binding to proteins, other RNAs, ligands, or metabolites. And although the secondary structure of the RNA has been extensively studied, its 3D structure is far from being understood. In this work, an attempt is made to understand the structural evolution of the RNA by doing a simple task of exploring the relative 3D orientation amongst the RNA's unpaired nucleotides. So around 13,000 RNAs whose structures were experimentally resolved were retrieved from RNA solo repository, and the unpaired nucleotides of each of those structures were identified with the help of the X3DNA DSSR software applied on RNA PDB files. So for each RNA molecule, the unpaired nucleotides were then retrieved and then clustered using DB-scan, and subsequently the unpaired nucleotides that were in the same cluster but had order of 1,000 nucleotides between their position on the sequence were filtered. These nucleotide pairs were called high-bent nucleotides, and they were basically close in 3D space, less than 40 angstroms, but far apart in the sequence, more than 1,000. The 3D position of a nucleotide was represented here by its C4 atom coordinates. So what was observed was that certain combination of nucleotides, namely combination of G and A and A, and also A and U, were overrepresented amongst the high-bent pairs, as you can see in the bar plot here. And also I looked at the relative orientation between the high-bent nucleotide pairs, well, first of all, the orientation of a nucleotide is defined by its three atoms, namely C4P and N1 or N9, depending on the nucleotide. And then the relative orientation between two nucleotides was defined in terms of two angles and a distance, psi, theta, and d, where angle psi was defined as the angle between the normal vectors of each of those planes that define each nucleotide, and angle theta was defined as the angle between the two C4P vectors of the two nucleotides, and value d was simply a scalar representing Euclidean distance between these two nucleotides, or namely their C4 coordinates. So interestingly, what was observed was that certain relative pairwise orientations were extremely rare in 3D space, actually, across all molecules. If you can see here in the 3D plot here, you can see that the white part represents a lack of data, where there was no pair of nucleotides that had theta of 90. And on the other hand, certain orientations such as of 70 degrees were prevalent. These are the red areas in this sphere plot. And basically what was observed was that as RNA gets larger and larger, its nucleotides naturally get closer and closer to each other relatively. And however, there is an uneven distribution of these relative orientations that incidentally occur, which suggests a natural tendency for the RNA to stabilize into conformations that favor certain positioning of its unpaired nucleotides against each other, which could be useful for RNA computational design in drug discovery as a component. Finally, one of the RNAs that contained many high-bent nucleotide pairs is this RNA virus. I hope I'm pronouncing it correct. Alastavi Arva RNA virus, shown here. As you can see, there were 6 nucleotide pairs, CU nucleotide pairs, that were 16 angstroms apart and had exactly an angle of 7